Hi, Mike here. So we're going to look again just how complex is life? Is it just by random chance? Now I've had a fair bit of criticism from atheistic evolutionists, and we're going to now look at this from the scientific point of view, from some scientists. So there we have a bug that has gears on it. So how does that evolve? How does it evolve that? Gears on bugs? Really? Is colourful beauty just by chance? We saw all this beauty in the world, and how could evolution do that? So let's see what just a few sensible scientists say. And there's lots and lots of sensible scientists who don't believe in Darwin, don't believe in evolution, because when you actually study and look at evolution deeply and look past all the just so stories, it's impossible. So if we or anyone looks deeply into DNA and its complexity, with the many eminent scientists like Dr. Rob Stadler or Dr. James Tour, so there they are there. So that's Rob Stadler, James Tour. Now, Dr. Rob Stadler, he wrote a book um, which was a scientific approach to evolution, and it becomes clearly obvious that DNA cannot evolve by natural selection and random chance mutations if you look at what the sensible scientists have got to say. It says there what they didn't teach you in biology. And he wrote this book, The Stairway to Life, which is an origin of life reality check. So we're going to have a look at that now. So this is a clip from him talking about his book, The Stairway to Life. So Dr. Robert Stadler. The full video I'll link in the notes below. So it says there's Carl Sagan, support for abiogenesis. The origin of life must be a highly probable affair. As soon as conditions permit, up it pops. Well, that's very scientific, isn't it? There's Jeremy England. You start with a random clump of atoms, and if you shine light on it for long enough, it should not be so surprising that you get a plant. Okay, we will let Dr. Robert Stadler talk now.
Fascinating. And then we have our friend Bill Nye, who wrote a book called Undeniable. And in that book, there's a quote: "The origin of life just requires some raw material that could allow the spark of life to emerge." This quote kind of reminds me of Walt Disney's movie Pinocchio, where you have a wooden puppet and it just needed the spark of life to turn into a boy. And I think it's almost that kind of imagination, I think, that is leading Bill Nye to believe this, although he would say it is a science-based statement. And then perhaps most disturbing of all is what our kids in school are being taught. In the public schools, in the biology class, here's a biology textbook that says, life began when organic molecules assembled in a coordinated manner within a cell membrane and began reproducing. And then another biology textbook, even more succinct, life arose from non-life via chemical evolution. And chemical evolution is a process where molecules reproduce, well, a hypothetical process where molecules reproduce themselves and have some errors, perhaps occasional errors in reproducing themselves, and maybe that occasional error led to a better molecule that's better at reproducing. And it's sort of a natural selection then makes for better molecules, the chemical evolution. So these two statements from biology textbooks are so disturbing because they exude confidence. There's not a hint of any uncertainty here, like we think life began, or maybe perhaps life began. They just say it very strongly. And this is what our kids are being taught. So what's really going on here? And this book really simply tries to answer the question of what would it take to start life by purely natural processes. And so Change 10 and I built this into a staircase or a stairway. We call it the Stairway to Life. And it's basically a set of requirements, almost like engineering requirements. We have to meet each of these requirements in order to advance up this staircase, starting from chemistry, starting from very basic molecules, very simple molecules, and building up to where you get to a life form, and then you have biology. So we're trying to get from chemistry to biology, and we have all these requirements. I'm not going to claim these are in the proper order. You know, I don't want to argue about the order, but the order doesn't matter. You still have to meet each of the requirements at some point, because we know life has all of these things, and simple molecules do not have all of these things. And we also know, as we already discussed, that there's almost like a gravity that pulls down the staircase. You know, we know that death and decay is a natural process that very easily, very readily always goes down the staircase. And we're going to talk about what could possibly get you to go up the staircase here, which is this hypothetical process called abiogenesis. So we're not going to look at every step in the staircase. We're just going to hit a few highlights, pull out some interesting parts. So the first one I'd like to look at is what's called homochirality. And homochirality, we'll see, is a very unnatural phenomenon, meaning it cannot happen from, prebiotic or just through natural laws. So some of you may know, but if you take an organic molecule that has a carbon atom with 4 different groups attached to it, if the 4 groups are all different, each one of them is different, then that molecule will have what's called chirality, which means it can be in a left-handed form or a right-handed form. And those 2 forms sure look the same, just like your hands do, but they're not superimposable. They're not the same. And if you produce them in a laboratory, you'll get a 50-50 mixture of the left and the right-handed. It's just natural because the reaction will randomly pick this kind or this kind. And so you end up with a 50-50 mixture because they're energetically the same. So it would be very natural if life contained 50-50 mixtures whenever you have a chiral location, because that's what natural processes do. But what we find in life is dramatically different than that. For example, in proteins are made up of amino acids. And here we have 4 amino acids hooked together in a chain. And almost every amino acid has a chiral location indicated by the arrows here. But we find in life that all of these chiral locations in all proteins are left-handed, always exclusively left-handed. And it turns out that they need to be left-handed because if only one amino acid was replaced by a right-handed, the counterpart, then the protein that you form would not fulfill its task properly. It wouldn't work because having the wrong chirality, reshapes the molecule, and it won't have this nice structure of alpha helices or beta sheets that are very common in proteins. And if you didn't know this fact, then perhaps you don't know Jack Schitt's, the name of the first author here in this. Very interesting name for a guy. But that's not all. We also have homochirality in DNA. So we have the DNA molecule here. You're all familiar with that. And DNA is made up of a bunch of nucleotides. This is one nucleotide. Each nucleotide, every nucleotide has 3 chiral locations. So you could have 2 kinds of stereoisomers here, 2 here, 2 here. 2 times 2 times 2 means 8. There's 8 possible chiralities in this molecule. But it turns out that every nucleotide in every DNA in all of life, including all 3.2 billion nucleotides in 1/2 of your DNA, all have the exact same chirality. And that's very unnatural. I mean, as far as something that a prebiotic situation through natural laws could produce. And it gets worse because in
So we'll leave it there. If you want to watch the full video, I've linked it in the show notes. But if anyone is genuinely interested in why evolution arising from non-living chemicals is absolutely impossible, watch the full video that I've posted by permission from Dr. Rob Stadler. It's quite long, about an hour, but he goes into detail from his book, The Stairway to Life, and it's fascinating. It's absolutely amazing the amount of complexity that we need to start life. And he explains the step-by-step requirements that are needed to create any biological life. Now, Dr. James Tour, he's one of the most famous scientists in the world, and he's had lots and lots of papers, sort of published, and he's got lots of patents out there. But anyway, he challenged the 10 most respected origin of life researchers, and this video is from about 2 years ago. But why did he challenge them? Well, he challenged them because the origin of life researchers manipulate their research and publish results to give the media and the general public the impression that their research has proved how evolution started. Biological life from non-living chemicals. Well, this is known as abiogenesis, and it has not been proved at all. So here's Jim Tor now.
I am going to take down all the content on my YouTube channel where I've critiqued origin of life on one condition. The condition is this: I am going to name 10 key researchers that have published key papers in the area of origin of life, and I'm going to give all 10 of you a chance to answer 5 essential questions that need to be answered for origin of life to be solved. What are these 5 questions? Well, they're the same 5 questions that I put up on a recent debate that some YouTubers have said have already been solved. Show me the prebiotic chemistry that would do this coupling.
This scheme is what James wanted me to write on the board.
If their answer is good, take their answer. I'd like to see you, as a researcher in the origin of life community, take their answer and present that as a solution. All 5 have to be answered to have a model for the origin of life. You just answer one, and I'll take down all my content. Well, the 60-day challenge has expired. I have offered 10 people who work in the area of origin of life to answer 5 essential questions, 5 of many questions that have to be answered in order to build a living cell. One of the questions had to do with making polypeptides, another one with making polynucleotides, the RNA, and another one having to make the polysaccharides. I gave them all of the enantiopure small molecules, all the amino acids, all the nucleotides enantiomerically pure, and all of the sugars enantiomerically pure. And I said, all you have to do is polymerize these. None of them could answer those questions. Then I had, what is the origin of information? And finally, I had, build a cell. Let's say you had all of the different components, all the polymers, all the small molecules, and all you have to do is construct a cell. None of them could answer those questions. It's not that some people submitted questions to the experts. No, nothing came in. Nothing. Remember I told you that I would take down everything on my YouTube channel? I am going to take down all the content on my YouTube channel where I've critiqued origin of life. And I'd never publicly talk about it again. If all they did was answer one of those 5 questions, I'd take down everything. I'd stop bothering them. They couldn't do it. They couldn't answer it. So I'm not going to take down anything. And remember what I told you, that if they can't answer any of the 5, I will stop saying that we are clueless, but I'll start saying that we are utterly clueless. The entire area of origin of life is an absolute scam.
Origin of life research is a scam.
Okay, it was tongue-in-cheek, but yeah, I think, and I meant it as tongue-in-cheek. Obsession with the identities that we see in biology right now are giving us a false sense of security about what we're looking for.
This is a huge field, and people just wander through it saying that they're working on the origin of life, when actually what they're doing is having fun with something that struck their fancy, and perhaps even if all the work goes exactly as planned, everything turns out well, they won't have actually learned anything about the origins of life.
The scam is, if we just make this RNA, or let's make ATP or ADP, we've got that part nailed. Let's now make this other molecule, another molecule. And how many molecules are going to be enough? And even now, today, synthetic biologists cannot make a cell from scratch because there's some contingent information embodied outside the genome in the cell. And that is just incredible. So there's lots of layers to the scam.
Even in their own laboratories, they can't put these things together. I gave them the 5 questions, which a YouTuber said have all been answered already in the literature.
I've showed you prebiotically plausible routes to all of those molecules and the assembly of a protocell.
No, the YouTuber is wrong. This is what I've been telling you. Here are the 10 experts who wrote the papers that the YouTuber cites. They are confessing that their papers don't answer the questions because they didn't have the answers. The only one to respond to me was Lee Cronin, and Lee Cronin kindly said that my questions were not relevant. The questions were not relevant for the origin of life. I'm not sure how that can be, but what he agreed to do is to meet with me. We were invited to a Harvard University roundtable. He and I are going to be at Harvard on November 28th, and it was very big of him to come to Harvard and have this discussion with me. There'll be about 100 faculty from Harvard and MIT that will be attending, and maybe from a couple other universities. Almost everyone in that room is going to be favoring the side of Lee Cronin, and that's fine. I said, I don't need a panel. I'll just talk to them myself. We will go through this together. We will have a discussion, and he's going to tell us how the origin of life came about. All 10 of those people were invited, and Lee was the only one to agree to meet with me.
The sequences of the amino acids and the proteins from humans.
Now, Steve Benner did write back. He said if Jim Tour had given me what he gave Dave Farina in that debate, meaning that all the small molecules in 100% enantiopurity, he would not have even needed slides. And in 1 hour, he would have explained how life could form. So I said, Steve, why don't you come up to Harvard and we'll discuss that? He said he didn't have time. Okay, he didn't have time. I said, how about if I come down to your institute? I will come down to your institute and you explain it to me. It's not easy to get there. You have to fly into Gainesville, Florida, which is a small airport. And then from there, you got to drive almost 2 hours to get to Steve Benner's institute. And I said, you can have everyone in your institute on your side and just explain it to me. Because you said you could explain it in an hour. I'll sit there for 3 hours and I'll come down on my own money and explain it to me, because you can do this in 1 hour. You could explain how life would form.
You can come back to a sort of an idea of what the essence of life is.
And his reply was, well, my health and my time won't permit it. So this is not a serious scientific discussion. I mean, if somebody says that they will appear in your office paying their own way, and all you have to do is explain to them something that you can explain in 1 hour, and then they decline, this is not a real discussion.
What I'm going to try and do in the next 15 minutes or so is tell you about an idea of how we're going to make matter come alive.
Lee Cronin, I'm excluding him from this conversation because he was big enough to say he will meet with me and we will discuss it. There will be a film of this made. It won't be live streamed, but we'll end up posting that. But as far as these others, nobody came with anything. This is a house of cards. This is what I've been telling you. And remember, these were just 5 questions. I could have asked them, how about the mass transfer problem? How much material would you need to go from A all the way to Z, this construction of a cell? How would you do that? How would you do this without any sort of relay synthesis, without keeping a laboratory notebook? We never talked about the Leventhal 1.0 problem, the folding of proteins. We never talked about the Leventhal 2.0 problem, which is the interactomes, the non-covalent interactions that have to take place between protein, DNA, RNA, all sorts of these interactions that need to take place. We have no idea how all these substructures within a cell would be synthesized. Of the substructures, do you know how many have been synthesized by any of the origin of life chemists? 0. So we don't know any of this. And these folks keep doubling down and they act as if they know. They'll say that we're right on course for Darwinian evolution to take over now. No, we don't know. We have no idea, and they'll stand by their primordial soup model, and they'll say this thing happened in a pond somewhere, next to the ocean somewhere, and these molecules came together and it happened. So that's the state of the origin of life field.
I haven't made life in my lab yet. I will.
There's one possibility. Lee Cronin may come on November 28th, and I hope he does. And he explains to us how life could have originated. So we'll give Lee a chance, and I really admire him for stepping up and doing this.
So Lee Cronin did meet Jim, and it's on YouTube. You can actually watch that. It's very good. Anything of James Torr's is worth looking at. So James Torr, he's a Christian, so's Rob Stadler, and so's Stephen Meyer and Douglas Axe and many, many scientists. So we'll move on. So Dr. Stephen Meyer, PhD, and he's the author of Darwin's Doubt, Signature in the Cell, and recently Return of the God Hypothesis, because we can now look at the science and see that it's too complicated to have happened by random chance mutations and natural selection. And then this video also has Douglas Axe in, who's a molecular biologist in the Biologic Institute, and he wrote a book, Undeniable, which is also worth looking at.
The Cambrian explosion is not just an explosion or the abrupt appearance of many new forms of animal life. It's also an explosion, or would have required a huge infusion or generation of new biological information. Biological form requires biological information.
Scientists' understanding of biological information advanced dramatically when Cambridge University researchers James Watson and Francis Crick made a startling discovery. They found that the structure of the DNA molecule stores information in the form of a 4-character digital code. Strings of precisely sequenced chemicals called nucleotide bases supply the assembly instructions, the information for building the crucial protein molecules that living cells need to survive. Crick later came to realize that the chemical constituents in DNA function like letters in a written language, or digital symbols in a section of computer code. Just as English letters convey a particular message depending on their arrangement, the sequences of chemical bases along the spine of the DNA molecule convey precise instructions for building proteins. The arrangement of these bases directs the arrangement of the 20 different kinds of amino acids that make up protein molecules. Proteins in turn perform a vast array of critical jobs inside cells, catalyzing reactions, processing genetic information, and forming the structural parts of molecular machines and other biological structures. Building new animals requires many new protein molecules, and building new proteins requires new biological information.
I used to ask my students a question when I was teaching. If you want to give your computer a new function, what do you have to give it? And they would know. They would say code or software or instructions, a program. All those are the correct answer. To generate a new function in a computer, you have to have code, you have to have instructions. The same thing turns out to be true in biology. This is the great discovery of the second half of 20th century biology, that information is running the show in biological systems. To build a new form of animal life requires cell types, requires proteins, and therefore requires genetic information. And that's the big question that the Cambrian explosion presents. If you want to think about how to build an animal, how would these animals get built, you have to have some explanation for the informational requirements of their construction.
According to modern evolutionary theory, new proteins and new forms of animal life arise through random genetic mutations, sifted by natural selection. But in an alphabetic text or a section of computer code, random changes typically degrade meaning or functionality and ultimately generate gibberish.
As we've come to appreciate the digital or typographic character of genetic information, it raises some really interesting questions about the efficacy of that mutation-driven mechanism. And we know from computer code, for example, that if you start making random changes to a section of computer code, you're much more likely to degrade the information that's there already than you are to come up with a new operating system or program.
This problem has long been recognized by computer scientists, mathematicians, and engineers, including a group from MIT who convened a now famous conference at the Wistar Institute in Philadelphia in 1966. These scientists met to consider whether the random mutation natural selection mechanism could conceivably generate enough biological information to build a new animal or even a new protein in the time available to the evolutionary process. One of these scientists was MIT engineering professor Murray Eden.
No currently existing formal language can tolerate random changes in the symbol sequences which express its sentences. Meaning is almost invariably destroyed. Murray Eden, MIT.
Eden knew that random changes to alphabetic or digital characters inevitably degrade the information in any section of alphabetic text or digital code, and for a very good reason.
Whether you're talking about digital code in a software program or a section of text in an English sentence or book, or the genetic text in DNA, there are vastly more ways to arrange the relevant characters that convey the information in a way that will produce gibberish than there are that will produce function or meaning.
Eden and his colleagues suspected that the genetic code faced a similar difficulty. When it came to producing new genetic information, at least enough to generate a new protein, the random mutation natural selection mechanism had to deal with what mathematicians call a combinatorial problem. In mathematics, the term combinatorial refers to the number of possible ways that a set of objects can be arranged or combined. In genetics, the combinatorial problem poses a severe challenge to the random mutation natural selection mechanism. To understand why, imagine a thief who would like to steal a beautiful new bike. All that stands between the thief and the bike is a lock with 4 dials, each marked with the numbers 0 to 9. But there is only one correct combination that will set the bike free.
The reason a bike lock works is that there are vastly more ways of arranging those numeric characters that will keep the lock closed than there are that will open the lock.
A thief without knowledge of the combination must guess the right combination from among 10 times 10 times 10 times 10 possibilities. That's 10,000 possible combinations, which usually would be more than enough to defeat a random search for the one right combination. Yet there is still a way the thief might succeed. If he has enough time to try enough combinations, he might eventually identify the right one by chance. For example, if trying each combination takes 10 seconds, then in 15 hours an especially diligent thief could try more than 5,000 combinations, or more than half of the total possible combinations. In that case, he would be more likely to succeed than to fail in opening the lock. But now imagine a much more complicated lock. Instead of 4 dials, this lock has 10 dials. Instead of 10,000 possible combinations, this lock has 10 to the 10th power, or 10 billion possible combinations. With only one combination that will open the lock out of 10 billion, it's much more likely that the thief will fail, even if he devotes his entire life to the task. So what about relying on random mutations to search for a new DNA sequence capable of directing the construction of a new functional protein? Would such a random search for new genetic information be more likely to succeed or fail in the time available to the evolutionary process? In other words, is a random mutational search for a new gene or protein more like the search for the combination on the 4-dial lock or the 10-dial lock? The scientists at the Wistar conference were unable to definitively answer that question because at the time, no one could adequately quantify how hard the search problem was.
So in the late 1960s, one could make these arguments based upon analogy to things we understood, written language, computer code, but we didn't have any experimental data to show whether those analogies actually suited the biological case. So no one could come up with the exact numbers to answer these questions back then.
Molecular biologists at the time did know that the number of possible combinations corresponding to any given sequence of DNA is extremely large and grows exponentially with the length of the molecule in question. For example, corresponding to one short protein 150 amino acids long, there are 10 to the 195th power other amino acid arrangements of that length. That's an unimaginably large number. But scientists in the 1960s didn't know how many of those arrangements were actually functional. They didn't know, in effect, how many combinations would open the lock. That didn't stop evolutionary biologists from speculating. Many argued that there must be a high proportion of functional sequences among all possible sequences, so that a random search for a new functional sequence would have a high probability of success.
The way they did that is to say, well, maybe biological sequences are not nearly as finicky, not nearly as picky about which characters are where as written language is or as computer code is. And so that's the track that they took. That maybe proteins don't really care which amino acid is where, and there's a great deal of variability, and therefore you can have the same function performed by a huge number of protein chains and a huge number of genes.
But recent experiments in molecular biology and protein science have replaced speculation with data. These experiments have established that DNA-based sequences capable of making functional proteins are in fact extremely rare among the vast number of possible sequences. Just how rare? After working at Cambridge University, molecular biologist Douglas Axe set out to answer that question, using a technique called site-directed mutagenesis. His experiments enabled him to estimate that for every DNA sequence that generates a functional protein of just 150 amino acids in length, there are 10 to the 77th amino acid arrangements that will not fold into a stable three-dimensional protein structure capable of performing that biological function. One correct sequence for every 10 to the 77th power incorrect sequences. That's equivalent to searching for just one combination on a lock with 10 digits on each of 77 dials. To put this in perspective, keep in mind that there are only 10 to the 65th atoms in the entire Milky Way galaxy.
So it's incredible, isn't it? The amount of chances the Earth would have to be, how many billion years old? 50 billion years old, 100 billion years old. It wouldn't be long enough for any of this to happen. Dr. Stephen Meyer is founder of the Discovery Institute, and of course he promotes intelligent design, which is the fact that, our DNA and all life on Earth was divinely created, because information can only come from a mind. There are only two options for all the DNA that is in all the beautiful life we see. Either DNA evolved by random chance, for which there's no empirical evidence, just wishful thinking and assumptions and just so stories, or DNA is the medium that the divine creator used in the very beginning to create all the colourful, wonderful, adaptive complexity of life. Unless serious investigation of the evolution theory is undertaken by individuals who might be really curious about how life started, and they'll only know if they start looking at evolution in a bit deeper. They'll never find the truth. The creator of all life, who created the DNA, gave mankind his message, his guidance, his promises of salvation for people who believe in and read the divinely inspired word in the Bible. I'd like to thank Almighty God and His only Son, the Lord Jesus Christ, each day for my life, for my family, and a promise of eternal life to come, and all the experiences of life that I've had. There is an amazing evidence for creation. You just have to look deeply and look beyond the fake news of Darwinian stories. The really good news is many young people now are seeing through the evolutionary deception. They're challenging the evolutionary paradigm in their schools and their colleges because that's all they're allowed to teach. They don't teach intelligent design or the fact that we could have been created. And this is because of the scientific progress. This is the reason why they're now challenging the evolutionary paradigm, because we've got a recent understanding of the complexity of DNA, which is being rigorously researched by the ENCODE program, which is the genome program. So they started with humans and then they went on to animals, and now they're insects and fish, and all the complexity of it shows us that life could not have evolved, but there are still millions of scientists who refuse to accept the reality of life. Even when they see the complex evidence, they do not want a creator. Check out the next slide. Here's one of them. In Matthew chapter 7, verse 7, Jesus said, "Keep on seeking and you will find. Keep on knocking and the door will be opened to you. For everyone who asks will receive, everyone who seeks finds, and to everyone who knocks, the door will be opened." There are thousands of scientists who hold this view. Evolutionary biologist Richard Lewontin explains his commitment to materialism no matter what. He has a prior commitment to materialism. It is not that the methods of science somehow compel us to accept a material explanation of the world, but, on the contrary, we are forced by our adherence to material causes to create a set of concepts that produce material explanations, no matter how counterintuitive, no matter how mystifying. And he goes on to say, materialism is absolute, for we cannot allow a divine foot in the door. We cannot allow a God, a creator, in the door. So lots of scientists, they deny the obvious and promote the story of evolution. It's so very, very sad. And of course, this is all gets put in the media and millions of people believe them. So this is Professor Anthony Flew, who was a staunch evolutionist all his life and used to really criticize anyone that believed in a creator. And he wrote this book recently, because he did say that he wrote a book, There Is No God, but now he's changed that to say there is a God. And if we look there is a God, how the world's most notorious atheist changed his mind. This is from his book, My Discovery of the Divine Has Been a Pilgrimage of Reason and Not Faith, meaning he saw the evidence for God in the fact that it's all too complex. It wasn't that he was led by, reading things. And it says there, it speaks very well of Professor Flew's honesty. After all these years of opposing the idea of a creator, he reverses his position on the basis of evidence. So all these scientists, there's many, many scientists who see the complexity of life, and they know and understand. Now, these are all Christians because they see that it couldn't have evolved. So you can go on YouTube and find this. So that one on the right, top right there, is James' tour, and it's an in-depth course for students, modern students today. It's 9 hours of content. It's very deep and complicated, but it's showing that evolution is out the window. The bottom left there, that's Michael Behe. He does a whole series on the secrets of the cell. If anyone watches that, you will know that evolution is bunkum. And there on the right, we've got by design, Behe, Lennox and Meyer on the evidence for a creator. It's intelligent design explained. It's God's creation. So if anyone really seriously wants to know the truth, you just have to look a little bit deeper, because evolution is just storytelling, assumptions and wishful thinking. So I'll say bye for now, and yep, I'll put another video up soon. Okie dokie.